I would request our chairperson, Dr. Mrs. Neha Kavathikar, ma'am, to come on stage. May I now request Shrika ma'am again to greet Dr. M. Scott Hall with a big key. Now I would request Dr. Mrs. Neha ma'am to throw light on the topic for today's first session. Ma'am.
Myself, Neha Kavathikar, welcome you all for this scientific session. I am pleased to be here and have a chance to introduce our guest speaker, Professor Scott Hall. He has been one of the main proponents of this international conference on recent advances in translation research and molecular medicine. His topic today is insight into the lethal toxicity of synthesis of cathinones from studies in larval zebrafish. He is Associate Professor and Director of the BSPS Pharmacology and Toxicology Program. He has completed his PhD in Neurobiology from University of Cambridge, UK in 1994. He has completed his postdoctoral from National Institute of Alcoholism and Alcohol Abuse from 1994 to 1999. After postdoctoral fellowship, he was funded by the National Research Council and National Institute on Alcoholism and Alcohol Abuse. He led a transgenic mouse research group in the National Institute of Drug Abuse in Baltimore. He was awarded by International Behavior Neuroscience Society recognition for its outstanding contribution and dedicated services. He was also elected as President of International Behavioral Neuroscience in 2016, President election 2016 to 17, President 2017 to 19, and past President 19 to 20. The Hall Laboratory investigates addiction and related psychiatric disorders, including attention deficit hyperactivity disorder, autism spectrum disorder. A major research focus has been upon the lethal and toxic effects of cathinones using mouse, zebrafish, and cell culture models. The current research of him in the laboratory focus on several aspects of such models. So his topic today is insight into the lethal toxicity of synthetic cathinones from studies in larval zebrafish. Without further ado, please join me in welcoming Professor Hall.
For inviting me here and for organizing this wonderful meeting. I'm going to talk about something a little bit different from what a lot of people have spoken about. A lot of people have been more focused on developing drugs, developing treatments for disorders. I'm going to talk today about addiction, and more specifically about drug overdose. So to begin with, I usually start all my lectures on drug abuse with a question. That is, why do people consume drugs of abuse like alcohol or nicotine or cocaine in the first place? And one of the things to think of is, firstly, most adults in pretty much all countries consume one drug or another. The vast majority of adults do not have problems with that intake. So if I could ask you, how many people drink coffee or tea? Majority of people here. It's a mild drug. How about alcohol? I'm not sure about the honesty there. Alcohol, try once more. I've had alcohol with some of the people here, so I'm sure that's the case. I usually ask about marijuana, and there start to be fewer hands as well too. People are, for some reason, are unwilling to admit that they use illegal drugs. And I would suspect in a room like this, I would get a smaller number of people who have used a lot of drugs. For the simple reason that the people in this room, all of the students here, have been very successful in your lives. One of the things about addiction is that it impacts the rest of your life. It's not so much about the drug taking. It's about how it impacts your life, how it dominates your life, how you lose control over taking the drug. So in the United States, the vast majority of Americans have tried 3 drugs. So nicotine, alcohol, and marijuana. Only a small percentage have trouble with that. So when we start talking about addiction, you have to start with that question. What makes those people different? So over my career, I've studied that in different ways. And there are obvious answers. So there's genetics. There's also environment. When you look at the reasons, more acute reasons why people take drugs, pretty much every person who has a problem with taking drugs suffers from anxiety or depression or PTSD. There are reasons for what they do. So to understand it, you have to start there. Now with drug overdose, it really comes from 2 different circumstances. Actually, is there a controller? Just do it from here, okay. There are 2 circumstances in which people typically have drug overdose. So one of those could be people who have a drug use disorder. And one of the things that characterizes that is that they elevate their consumption over a period of time. Taking so much of the drug, their body tries to compensate, and it changes. And it fights against the drug, essentially. So what happens is they have to take more drug to produce the same effect. And as you take more and more of the drug, you run the risk of an overdose. So that's one circumstance. The other circumstance is people who are relatively naive to the drug. They go out to a club, the friend says, try this. They take it, and it's too much for them, and they overdose. So there are a couple different circumstances in which that occurs, and we need to sort of bear that in mind. The circumstances that lead to overdose are different for different individuals. So, in the United States, this has been a big problem over the last 20 years. We generally refer it as the opioid epidemic. So it began with prescription opioids. And I was a member of a number of societies in the early 1990s as these drugs were being approved, and we all told everyone, the medical profession, the government, this will be a problem. You have to be very careful about this. We understand the need. We needed those drugs to treat pain. They were very important. But if you give them to millions of people, you will have tens or hundreds of thousands of drug addicts. Not a terribly good word. That's an old word because I'm old. But people with drug use disorders is a better way to phrase it. But in the late 1990s, early 2000s, they started to clamp down on those drugs to restrict their availability. And what we had was a second phase of that epidemic. As a lot of users switched to illicit drugs, they started using heroin because that was easier to get. And then the next big important phase was fentanyl. So there started to be the synthesis of illicit fentanyl. So this is not fentanyl like you get for pain. This is illicit fentanyl and other fentanyl analogs. And the thing to bear in mind about that is fentanyl is 500 times more potent than heroin. So this is what really ramped up the opioid epidemic. And that last rise there, coming up to last year in the United States, 100,000 people died from illicit drug overdoses. And that's just the people who died. There were lots of other people who suffered very serious consequences because of overdoses as well. Now, lost into that is one more thing. In the last few years, there's been a precipitous rise in overdoses from an amphetamine-like stimulants. So the classic amphetamines that have been abused throughout the world are methamphetamine, which is a stimulant drug. It produces excitement, euphoria, provides you with lots of energy, and MDMA, ecstasy, methylene dioxymethamphetamine. It's very, very different effects. It's not really addictive in the way that methamphetamine is. It's abused, but it's not addictive really. You don't start taking more and more of it. However, there have been overdose problems from these drugs for a long period of time. Over this same period, there's been the development of a large market throughout the world for what we refer to as novel psychoactive substances. So these are drugs that are similar to these others in structure and effects, and they were developed largely as a part of research programs trying to find new medications. And the development of a lot of these drugs coincided with the development of the internet. So the internet is very positive in a lot of ways, but it also releases information very widely. You can find any information you want. If I had wanted, when I was in high school a long time ago, to synthesize methamphetamine, it would have been difficult for me to find information about how to do it. Now I can just type it into a computer and the synthesis comes up on my computer in minutes. So this has changed the way drugs work to a large degree. So the drugs I'm going to talk about now are really drugs that are replacements for these classical amphetamines and stimulant drugs. A large number of them are based upon a chemical from a traditional plant. So the cat plant, which has traditionally been used in northeastern Africa and the Arabian Peninsula, but spread to other parts of the world as well, was traditionally used like a lot of other plants. It was chewed, it was drunk in a tea. The effects were relatively mild. But something that typically happens when a drug like that moves into more technologically advanced societies, when it comes to India, when it comes to the United States, when it comes to Europe, the first thing we do is we isolate the active ingredient. And that's what they did. The active ingredient is cathinone. It's the beta ketone analog of amphetamine. So it's one oxygen away from being amphetamine. So very much like amphetamine. And then people started making variants. And they've actually made around 300 variants. Around 300 variants have been found to be abused by people. So this creates a very difficult problem. How do you study all of these drugs? There's so many of them. There are dozens that are still emerging every year. We haven't been able to keep up with understanding what potential do they have for abuse, for addiction, for toxicity. When I moved to Toledo, I just published one of the first papers on the lethality of these drugs. And I looked around and I said, well, lots of people are studying the addictive properties. Somebody should study the toxicity, the lethal potential of these drugs. So I started a group that really focused on that. However, before I move on, I want to address this question a little bit. So one of the things that you might think is this is an American problem. We don't have that here. We don't have that. There are drug abuse problems everywhere, including in India. So when the first day I got here, I started just looking through the internet for stories, and one of the first things that popped up was that India is a major manufacturing center for chemicals overall. It's one of those places where you have a lot of contract chemical companies. You can hire them to make chemicals for you. And for about the last 10 years, India has been a center for making these cathinones. Because remember, or maybe not remember, I'm not sure if I said this, these are novel drugs. So one of the implications of that is they are initially not illegal because nobody's seen them before. Nobody thought to make them illegal. So initially they can just be sold over the internet in gas stations anywhere. So India became a center for making a lot of these drugs. But there are also a lot of illegal drugs that are made by some laboratories in India as well. Here's evidence of a large seizure of ephedrine. Why is that important? Ephedrine is the precursor for MDMA. That's why they very carefully keep track of it. MDMA is also being made in large quantities in India as well. There's been a rise in the use of these drugs in India. Simply them being made here makes them available to people. So they get sold here along with everything else. There's been seizures of amphetamine-like stimulants, cat leaves as well, and raids of some of these clandestine labs have seized large volumes of drugs. Methadone is the chemical on the right there, and it is very similar to methamphetamine or methcathinone. It has very similar effects. So as I said, most of these drugs were legal. They were legal in the United States, depending on the drug, most of them until about 2018. The newer ones aren't illegal until somebody makes them illegal. Although we have what we call an analog act, which means that anything that looks like something that's illegal is automatically illegal. However, it's very hard to enforce, because how do you define similarity in that sense? And I've had a concern in this as well. The original reason most of these drugs were developed was they had potential as medications. I don't want to see all these drugs banned because some of them may be useful to medicine. Bupropion is actually a drug from this class. So when I first started working in this area, there were a lot of media reports about how horrible these drugs were, the crazy things they made people do, how dangerous they were. And as I read those stories, I got very suspicious. The media likes to get people to listen to them. So they go for the most interesting stories. They don't always go after the most truthful stories. I'm sure it's different here, but that's the way it is in the United States. But this reminded me of something which was a government campaign in the 1950s in the United States over marijuana, where they were more concerned with influencing people than the truth. And the biggest problem with that is your goals may be noble, but if you try to influence people with lies and they figure it out, they simply stop believing you. And this was a major problem in the United States for a long time because everyone came to distrust the government on drugs. When you look at the clinical effects of overdose from all of these new stimulants, the synthetic psychoactive cathinones, their effects look like the classical stimulants. They activate you, they increase your heart rate. At higher concentrations, they can produce hallucinations, delusions, and ultimately they can produce coma and death as well too. Before that, they can produce immediate cardiac events, strokes, other things as well. So these new drugs at least have this potential, but we don't really know. It is very difficult to study humans. I can't take 100 of you, give you a drug, and see what happens when it's something like methamphetamine. They frown on us giving methamphetamine to undergraduate students. That's why we do animal research. It's a replacement for that. So a couple of the points to make. In terms of how much of a problem this is, we don't really know for a variety of reasons. A lot of these new chemicals are hard to measure. We haven't developed techniques to measure them in blood from victims, when identifying the powder when it's seized, in wastewater. We don't really have methods for doing all those things. So we don't necessarily always know how much of them are being used. And the people taking them don't know either. So this slide shows the results of a study that was done in New York City. So a lot of these drugs are used to replace MDMA, and they are called club drugs, because people tend to take them when they're going out to bars or clubs or concerts, social engagements. And a lot of times people will be sold these saying this is ecstasy. So they take it thinking it is MDMA. So what these researchers did is they went to clubs in New York City and they asked people when they were entering, have you taken MDMA in the last 3 months? And if they said yes, they said, can we have your hair? So they took a few strands of their hair and they analyzed it. And what they found was 60% of the people had taken these drugs unknowingly. They thought they had taken something else. Instead, they had these: belone, methylone, alpha pyrovalerone. A large number of cathinones were found in these people's hair. And most of these, we don't really understand what the dangers of them are overall either. So I started in the traditional way because that's what I do. I do mouse research as models for addiction or alcoholism or psychiatric disorders. Again, I said there's always this overlap between those. And my student Don Muskowitz started this work. So we did just classical LD50 studies. One of the things we looked at was temperature, however, because most of these drugs elevate temperature. Methamphetamine, MDMA, classically that's one of the main effects, and it's associated with their lethality. Well, the first thing we found was these cathinones did not increase temperature, they decreased temperature substantially. But I think this is a little bit of a mistake. People think that MDMA and methamphetamine elevate temperature, but part of the reason for that is the conditions under which they are tested. So if you test them in a warm environment, your temperature goes up. If you actually test them in a low temperature environment, the temperature goes down. What they really do is disrupt your ability to maintain temperature. So, but all of these drugs profoundly affected temperature homeostasis. So in some cases, the temperature dropped by 8 degrees Celsius, body temperature dropped by 8 degrees Celsius, which is not too bad. You can stand an 8-degree drop in temperature. You can't really stand an 8-degree increase in temperature very well. But then when we looked at the lethality, they weren't lethal at all. We had trouble killing a mouse with these drugs. It was very surprising. There was some evidence of toxicity in the liver, in the heart, but overall, these drugs did not seem to be very dangerous. So this raises the question of where do those reports of that lethality come from? Well, there could be some of these that are more lethal than others, and we just didn't study the right ones. It could be that it requires multiple dosing. It could be that some drug interaction is important. Like I said, people take these and they go out to clubs. So they are drinking alcohol, they are smoking cigarettes. That might be important for that. Some individuals might be more susceptible for some reason. That's true of virtually everything. Temperature might be an important effect, and we're going to return to that in a little bit. So my graduate student, Yu Chen, continued this work, initially in the mice. And while he was doing his PhD, we made this transition to zebrafish, which I'll come back to in a little bit, but I'll just want to mention a couple of his findings quickly. So first of all, we elevated the temperature and all of the mice died. So these, we have a graded effect here on lethality with dose, and then we elevated the temperature and just all of them died. So temperature is a very, very critical effect. This is the first time that it had been shown for a cathinone. It also potentiated the toxicity that we saw in the organs, just in terms of weights, but also in terms of histology. We had the typical effects of lowering temperature, but one of the things there is we were studying the animals that survived because we were throwing out the animals that died because we couldn't measure the temperature in animals after they died, really. But what we did, interestingly enough, was we measured the temperature at the point of death. So right when we noticed the animals died, they were sort of continuously being monitored. We took their body temperature. And those animals that were in low temperature conditions, their body temperature had actually dropped. The ones that were in the high temperature conditions, their temperatures had gone up. So again, it's this disruption of temperature homeostasis more than just hyperthermia per se. So the problem with using mice to study these drugs is that it was very slow, expensive, and time consuming. So something like, for a long time, Dr. Williams was saying 15 years ago, I started the zebrafish laboratory. I think he said that for like 10 years. So I think it's more like 25 years that we've had the zebrafish at the University of Toledo. And most of the researchers actually in our department have started to incorporate zebrafish into their work. And there are a number of reasons for this. For me, the most important thing is in the center of that slide, there's a 96 well plate. And it's very small, you can't quite see them, but each well contains a zebrafish larvae. They are 5 days old. So on that one plate, we can do an entire lethality assay in 2 hours. So this allows us to greatly accelerate the pace of our work. And we can also do a variety of other assays in this high throughput manner that help us to understand the mechanisms underlying this toxicity as well. So one of the other big advantages of zebrafish, one of the reasons we chose them is, particularly larval zebrafish, is you can see through them. You can see the internal organs. So we can simply look at the heart and take a video image and analyze that video image, and we get something that looks essentially just like an EKG. It's not based on electrical activity. It's based on the volume of the atrium and ventricle, but it looks exactly like an EKG. And if, in some respects, effectively is. So when we looked at the cathinones, we found very disturbed cardiac functions. Is clearly an important aspect of this. Another one of my recent master's students looked at the other question, which was the interaction with other drugs that people commonly take along with these. So firstly, he looked at ethanol, and ethanol increased the lethality of MDMA, and then also methylone, one of the classic SBCs. It's the beta ketone analog of MDMA. But then he looked at nicotine, and it produced a different effect. So we were expecting that it would increase the lethality. Partly I thought this because one of the effects that nicotine has is to elevate blood pressure as well. So I thought that would interact and cause increased lethality, but it didn't. It was protective, substantially protective against both MDMA and methylone. The converse was actually not true. So at the bottom there, MDMA or methylone didn't affect nicotine toxicity, but nicotine affected their toxicity. So that actually is a very potentially important finding. And the reason for this is, if you think of opioid overdoses, we know exactly what to do. We have opioid antagonists. But for stimulant overdoses, we don't actually have any treatment. It's all symptomatic. If you have high blood pressure, we try to reduce it. If you have a high temperature, we try to reduce it. If you're delusional, we give you a benzodiazepine. But we don't have any actual treatment for the toxicity. So this would be a very important advance if we could manage that. Now, in terms of understanding that toxicity, this is another paper from a couple years ago now, from a student at Dr. Schaefer's laboratory, using his technique. So this is photo affinity labeling. So what we did is we created a drug that looked very much like methamphetamine. It was modified in a couple locations. So a couple things happened. First of all, you shine the right frequency of UV light onto the animals after administering this, and it attaches to the closest protein. And then you shine light on it, and it will fluoresce. So you can use this to select out the proteins that are interacting with this molecule. So this molecule has effects that are very similar to methamphetamine. What we ended up studying primarily was not really the stimulant effects of the drug that you have at low doses, but really the depressive effects, the impairing effects that you have at high doses. So we were really studying toxicity, which I was quite happy with because it fit into my other research. I think it'll be important to study the lower doses as well too, but we're working on that. So what you get is a series of bands where you have this molecule interacting with the proteins. You do proteomics and you identify all of those proteins, and then you look for patterns. And that's what's represented in the bottom, is the network of interacting proteins, the systems that were affected primarily by this drug. And what it seems to be doing is impairing mitochondrial function, and that may be fundamental to its toxicity. So again, this is one of our directions for future research. So overall, what I want you to take away from this, more than anything else, is that the zebrafish larvae approach can really contribute to this area of research substantially and advance it far more quickly than some other approaches. Now, I'm not saying that I will not use mice. I'm not saying I will not use cells. I want to do all of them. I want to use people too, although not necessarily giving them these drugs, but studying overdoses. That combination of things will produce more advancement, I think, than anything by itself, but the zebrafish have an awful lot to contribute. And I would argue that it is a valid model, because many of the things that we saw in the mice, we also saw in the zebrafish. The effects of temperature, the evidence for cardiac and liver involvement in the toxicity, something I didn't talk about too much, but evidence for the involvement of ammonia and glutamate in the toxicity as well too. Now, there were a number of novel findings in this as well. So we didn't just validate what we'd already gotten in the mice, but we showed a number of novel things. So first of all, the lethal toxicity of SBCs is exacerbated by ambient temperature. That range of SBCs has never been studied in mice previously as well. Just the fact that zebrafish show this elevated toxicity in response to higher temperatures was novel. A lot of people didn't think that would happen. They didn't, because fish are cold-blooded. They thought they wouldn't respond in the same way that mammals do. But I think, in fact, they were more sensitive to temperature because of that cold-bloodedness. But we saw the same effects. And then we saw that ethanol exacerbated these effects, but nicotine is potentially protective. So this offers an avenue to try and really develop a treatment for the first time for stimulant drug overdoses. So what do we do next? Well, we really want to study broader structure-activity relationships. Like I said, there are around 300 of these synthetic psychoactive cathinones out there. So far, we've studied 9. So we have a little ways to go in terms of that. And one of the big issues there is getting them. We need chemists to synthesize them for us. And the other thing in terms of moving this forward is we really want to understand those nicotine effects. Will this lead to potential treatment that will prevent not just the overdose, but potentially some of the long-term consequences of the overdose for people who survive as well. So this work was done at the University of Toledo in my laboratory. The primary students who really worked on it is my former graduate student, Yu Chen, who's moved on to a postdoc. Current graduate students, Don Muskowicz, Sidney Russell, Logan Coons, and a couple master's students, including Abdul Rahman Bakanaid. And of course, Dr. Williams started all the zebrafish work. Isaac Schaefer and his students have synthesized all the drugs for me. And I, of course, want to thank our funding agencies, the National Institute on Drug Abuse, University of Toledo has been very supportive as well. And then thank the organizers and all of you for listening. Thank you. So if I did go too horribly over time, I forgot to look over a button. I can take some questions. To your body, one of the things it does is compromise liver function. And that has been shown in some of the interactions with amphetamines with temperature, is that what happens is that the liver is, to some extent, fighting against the drug. So it's metabolizing the drug and preventing some of its worst effects. And then at the higher temperature, what it does is compromise the enzymes in the liver that are degrading the drug. So we should be able to see that if we can measure the amount of drug in the 2 temperature conditions. But we haven't done that for the cathinones yet. That has been done in mice and rats for the amphetamines. All right, we have a microphone now. Oh.
One quick question. Okay, you can repeat my question.
Yeah, I'll repeat it.
Can you comment on the temperature effects on zebrafish? They normally they're present in water. Not what? I assume that the temperature of water is below 30 degrees Celsius.
Trying to remember. I think it's 28, 32.
So what is the maximum temperature?
Okay. So couple questions in there. First of all, there's a question about the cold-bloodedness, and this was sort of one of the concerns from the very first grant review I got on this, by the way, was that they didn't feel that the zebrafish would respond in the same way to amphetamines that mammals do, because typically fish are the exact same temperature as the water surrounding them. However, the thing to bear in mind is that they do not adapt to temperature changes very quickly. So this is actually, in fact, one of the places where they identified heat shock proteins. So you put fish in, you shift fish quickly from normal temperature water to warmer water, even just a small increase, and it will immediately affect them. They can adapt to very wide temperature differences over a season, where the temperature is changing over a long period of time very well. But quick adaptations, they are very poor at. It produces physiological stress. So that's part of that. The other question was about what the temperatures were, and this is off the top of my head, so it'll be accurate.
20.
Okay, well, that was going to be my guess. Thank you. I believe that the standard temperature is 28 degrees, and Dr. Williams confirmed that I was correct. And then the higher temperature that we were using was 32 degrees Celsius. Yeah.
Thank you.
All right. Oh.
Hi, Professor. My question is, how did you administer these drugs?
Okay. Yeah, I skipped over most of the methodological details here. So the question is, how do we administer the drug? So in particular, in larval zebrafish that are about that big, we're not picking them up and injecting them, obviously. So the drugs are administered by immersion. So we immerse them in the water. They absorb it relatively quickly. When they're very young, they're pretty much absorbing it throughout their body through the skin. And we do adult studies as well. As they get older, it's primarily through the gills. But their skin sort of changes over time as well, too.
So then comes my second question. So when you give ethanol along with the drug, is it because of good aqueous solubility, the toxicity has exacerbated?
So are you asking if the ethanol was altering the absorption of the other drug? We don't know. That's a possibility. I don't think so. I think it's additive effects, but the simple answer is that's the first study, so I don't know yet. So. All right.
So
any other quick questions? Oh,
quick question. When you do the protein labeling by photochemistry using MAP, I see two functional groups together, alkyne and azide.
Mm-hmm.
Do you see before getting level protein they get cyclized?
No. As far as I know, we don't see that.
Because I see the 5 carbon, you have alkyne, you have azide.
Yeah, I don't believe so, but I would refer you to Dr. Schaefer to give you a better answer. All right. Anything else quickly? Anything from the students? I usually get all the questions because I do drug abuse. Students tend to love that for some reason. I don't know why. All right. Oh, please.
I have, sir, increased temperature is developing, increase the toxicity
Yes.
of drugs. So similarly, those who are taking tropical region, those human beings are taking these drugs as in tropical region or subtropical African countries. So because temperature difference is there, can we draw this conclusion from that?
Yes, that's a great question. That's something I've really, really wondered about. And I was curious on coming here because I didn't know too much about drug abuse patterns in India overall. I knew about some of the drugs that had classically been used, but in terms of more recent drug use, I didn't really know. And it's something I've thought about because when a lot of the club drugs started to be used, in particular ecstasy. The places they were being used was Northern Europe, Northern United States. So I kind of wondered, was there a reason for that? Because they were colder countries, and maybe they were safer to use there. And I really wondered if these drugs were used in other countries that tended to be much warmer, would they have more lethal effects? I don't know the answer to that. I'm sorry, but it's a very, very good question overall. And particularly, again, as the world is changing, as the world's becoming warmer, we have to consider these sorts of things. Are those things going to interact with these things to make drugs more lethal? I don't know.
Thank you, sir.
Okay. Probably another question. All right, thank you.
Now it's time to greet you with mementos. So for this, I would request Dr. Rijwan sir to be on stage. Dr. Rijwan sir. Our next memento to Dr. Mrs. Neha ma'am. And our next memento to Dr. Amit K. Tillore sir. That will be given obviously by Dr. Piyush Tillore sir.
It has to be different.